Thanks, everybody. Yeah, so Island Pharmaceuticals, as opposed to the previous story, which I thought was very sexy, by the way, which was very broad, classic niche generic strategy. We're laser focused on an area of pharmaceuticals that is a little bit unusual, and it's driven by our strategic filters and also our network of advisors and people that we have linked with the company and the history of the company. So what we're doing is we're targeting viral threats and developing medical countermeasures. And maybe that's still not clarifying things for you very much. In addition to that, we really only like working with molecules that have a history, usual disclaimer rubbish, which I don't know why we have because it's an Americanism. So what have we got and what do we look for? So we've got known small molecules with a clinical history, and needless to say, if it's got a clinical history, it's also got a preclinical history, and the CMC, the manufacturing piece, is already done. So we're not interested in pulling things off the bench. We want to stand on the shoulders of giants. Mid to late stage clinical or abbreviated routes, and the abbreviated routes in the case of our new asset is incredibly interesting, and it's, to my knowledge, the first time an Australian company has taken this on. Open US FDA INDs, which means that the FDA has looked at it, likes it, has approved it to go into the clinic. Big tick. Validated US government military funding support is really important to us because we like spending other people's money, and PRV eligible. And if people want to go into PRVs, it's a different topic. But in essence, it's a way for the FDA to incentivise poorly served diseases. When you get your first drug approved, they give you a voucher for an abbreviated approval for a second drug. Doesn't have to be at all related. Weirdly, they're tradable. So small companies like us sell them. They sell every quarter routinely for $110 million. So as soon as you get your NDA, you get a PRV, you can flog it. So our strategic checklist is kind of a little bit of double up here, but really the last stop point is what I want to stress as one of our key strategic advantages, and it's not just the drugs we're working on, it's who we have working on them and who's in our networks. So between our Australian advisors and our US advisors, we have people with decades and decades of experience in military and medical countermeasures medicine. So one of our advisors, for example, was 22 years head of infectious diseases for the US Army. A very close relative in my household sits on the National Medical Countermeasures Task Force, et cetera, et cetera. One of our other advisors is on DMTC, which is Australian equivalent of BARDA. So I guess putting all of that together, we're uniquely, almost in the Australian context, set up to deal with these sorts of projects. And so why have we got these two molecules now? Why in particular have we got the second one that we're just embarking on due diligence on, but it's looking really good so far. Both these assets have completed phase 1 with a really good safety profile. Both have open INDs. They're both antivirals. We really focus only on antiviral drugs. It's a very specific medicine, a very specific area of drug development. Both of these programs have had appreciable or extensive US government support from a number of agencies, so not just BARDA, but also NIAID, congressional discretionary funding, a whole lot of buckets of money that we've identified in concert with these. It means we don't have to spend as much of our own money on these. And PRVs and multiple shots on goal for those PRVs or for indications. So both of the molecules that we're looking at have the ability to treat multiple viral diseases. So our core program, which we completed a phase 1 confirmatory formulation study for the FDA at the beginning of this year. We'll be going into our phase 2 with that within the next 10 days. We'll be dosing subjects before the end of this month. And I'll come to the description of that program as well, because it's slightly also a bit unique in the Australian context. And then that's our phase 2A, which is a prophylactic study, a preventive study. We'll be then rolling it straight into a phase 2B early in the new year. So a therapeutic study, so prophylactic then therapeutic. Effectively, the company will have executed 3 clinical trials in one 12-month period, which is pretty staggering for a small company. Our core program, clinical program, is targeted at dengue. We do have activity in other mosquito-borne viruses, but it's dengue that we're focusing on to take through into the clinic and is that we have government funding for, largely, again, US military. It's probably one of the biggest infectious diseases in the world. That's the headline case numbers. About 10% of those will end up hospitalised, and about 10% of that 10% will go on to develop a overreaction to the disease called hemorrhagic fever that is life-threatening. So why do we like ISLA 101 and why do we license it from Monash University? It's been through 45 clinical trials with J&J and the National Cancer Institutes for Cancer. Turns out safe as houses, not a very good cancer drug. We've recently completed a SAD study confirming our EC50 dose. That's the dose that is virally suppressive in multiple animal models. And those animal models were very severe. They were lethal animal models. And so we know that the EC50 dose that we're taking into the clinic in a week and a half's time to achieve the blood levels we want was capable of protecting rodents from lethal infection. As well as dengue, we can treat other diseases such as yellow fever, chikungunya, West Nile virus, a whole host of other mosquito-borne infections as well. So there's potential for label claim extensions down the track, either in our hands or a partner's hands. Multiple PRV options. Most of those diseases are PRV eligible. And we have a collaborative research and development agreement with the US Army where we're sharing data and receiving money from them. Phase 2 screening is underway. It's well underway. I think we've got them all screened now. So dengue's historically been thought of as a tropical disease, and most years still it is. That said, there's usually hundreds of cases up in the northwest of WA. They're usually fly-in, fly-out workers who live in Bali, but there have been community transmissions in North Queensland. There's routine community transmission now in Florida and the other Gulf states. And the Paris Olympics just passed because we're sort of party to some of this fury behind the scenes for medical countermeasures. They were terrified of that becoming a dengue super spreader event because they had hot, wet, humid weather and they had people flying in from all over the world, potentially carrying subclinical infections. Now, didn't turn out that way, but you should have seen them scrambling to get on top of that. So what are we actually doing in our phase 2A and phase 2B studies? I've kind of given you a broad outline. Primary endpoints is probably the main thing to just stress here. Because we're treating disease, we're not looking at surrogate markers, as you would in a cancer study, which are kind of, sometimes a bit wobbly. Our primary endpoint for both of these studies, the prevention and the therapeutic, is for viral suppression, so that we just don't see as much viral replication in the blood. The target organ for dengue infection is actually white blood cells. Secondary to that, we're looking at symptomology as well, so pain, rashes, diarrhoea, fever, clearly, and we'll be looking at all of those. We've also got another $1 million grant behind the scenes from the US Army, directed by Congress, to look at a whole host of other serology that's not really part of our endpoints, but it'll build a really compelling clinical history. So this is the structure of the two studies. Why is one preventive, one therapeutic? In a nutshell, preventive one, we dose them for 3 days and then infect them with virus, because I haven't mentioned to you so far that we're actually experimentally infecting volunteers with dengue virus. It is a slightly weakened dengue virus, but it's dengue virus. They will get sick. We want them to get sick because we want to see a suppression of disease. If they get too sick, they then go to hospital. We then follow them out for 20 days of therapy, and then we have follow-up continuing after that point, but we'll be doing a data read for the phase 2A probably mid to November. And then the therapeutic study, as you might imagine, we infect these candidates with virus first, wait a couple of days, and then we start treating them. So I guess that mimics a real-world situation for a treatment of disease a bit more closely, and the FDA wanted to see it. It opens up a second market for us. So we've got a prevention market for military, NGOs, travellers. We've got a therapeutic market for in-country infection, military again. So that's two separate label claims. My CEO loves this slide. He's a scientist. In a nutshell, what we're looking to do, that big hump in the middle, that's virus in the blood. We're looking to flatten that curve with our drug. The ones down the bottom is the duration and severity of the symptomology we're looking at. So again, I mentioned those before: diarrhoea, rashes, fever, pain. So our second program that we're in the process of acquiring, we announced a week or two ago, that we had signed a binding letter of intent to move into due diligence for this asset. This is an asset called Galidesivir that was developed by a NASDAQ-listed company called BioCryst. They had been developing it for two diseases called Marburg and Ebola. Probably you've heard of Ebola, you might not have heard of Marburg. They're very similar. Case mortality rate of about 90%. You bleed through every hole in your body until you die. Ebola in particular has already been weaponized. So the US Army is quite excitable about Ebola because they know the state and non-state actors who have dirty weapons. They also have dirty weapons containing anthrax and smallpox. There's already therapies for those two. There's not for Ebola yet. A lot of work has also been done in Marburg in monkeys, and I'll explain why that's important for us in a minute. And Zika is the other disease where they've got data as well. Now it happens we've got data in Zika with ILA 101 as well. We'll just have to make a line call as to which is the better drug for the Zika. So it's been through 2 human safety studies, this drug. It's been in the core programs in what's called the filoviruses, those hemorrhagic viruses. It's been through 3 what are called non-human primate studies. Unfortunately, when you don't want to start infecting people with Ebola and Marburg, you've got to start using apes for obvious reasons. They proved to be successful. Why is that important? This is a really unusual program, and there's been 15 drugs approved by the FDA down this route, but there's a potential for us to confirm and then move this development program down the route called the animal rule. And quote unquote, for diseases that are unfeasible or unethical to test in humans, you can potentially get a drug approval based on monkey data. So you do the phase 1, you do enough confirmatory animal studies to satisfy the FDA, you get a drug approval. And in this case, you also get PRV again. So as a best case scenario, if the FDA is still saying that we will only need to do primate studies and not human studies, we could potentially be filing for approval in 2026. We've already got 1 study that we're buying that's completed. BioCryst doesn't really like us talking about it in writing, but this program has had somewhere between $80 and $100 million US spent on it by the US Army. And as I said, it's PRV eligible. So what are the commercial opportunities for these drugs? Because they're not the same as what we just heard from our colleague here before, where he's selling through chemists, getting reimbursed for some of them, OTC for some of the others. These will largely be traveller and stockpile drugs. So the major markets are going to be the NGOs, travellers into endemic countries, particularly for dengue, not so much Ebola and Marburg, and then stockpiles for civilian and military defence. National outbreaks probably served by that same market. So we've already had unsolicited approaches from countries like India and Brazil asking when we might have drug available for stockpiling. Again, this is for ISLA 101. And again, both of them are PRV eligible across multiple diseases. So here's a snapshot of where we're at. Graph's finally heading in the right direction finally, that we've got some clinical trials underway. Quite clearly, if you look at our balance sheet, we'll need to raise some money at some stage in the not too distant future, but we have some sources of capital lined up that we are reasonably happy with. Change today on the board. So a gentleman called Dr. David Brooks resigned to pursue racehorses, basically. And Chris Terminopoulos from Perth has joined the board as of this morning. He's going to be a real asset to us moving forward with assisting with capital management strategy. And we're very happy to have him involved. Major shareholders, Jason Carroll, interestingly, he wasn't involved in the IPO. He's bought all of that on market. Bill Garner was one of the founders. I've known Bill for 15 or 20 years. He's a good friend. And Al Hansen, who's also on the board, as you see to the left, is the third biggest shareholder. Reasonably tightly held stock. Liquidity is going in the right direction at the moment, thankfully. And we've got a number of other grant programs to support our capital needs. as well that we've applied for recently or are going to apply for. We've actually just been invited to apply for a grant, just us, for Ebola by the US Army. So key milestones, as Stuart said, we have kicked some goals over the last 12 months. What have we got coming up in the next little while? Screening patients in the phase 2A is done. We had to get 2 lots of ethics approval because the Army owns the virus. We had to get it from the hospital where the trial's being done, so that's a bit unusual, but we've got all of that done. We'll be dosing subjects with drug and then dosing subjects with virus in the coming 2 weeks. Data readout from the trial will be before the end of this calendar year. I've already pulled together, and I'm driving this program, the due diligence team for the Galidesivir assets. So as well as the patents and the clinical history and the FDA correspondence, we're also buying truckloads of drug, which is always very helpful. We won't have to remanufacture. And this drug is very stable, so it will last probably through the entire clinical program. And again, we've got ongoing discussions with potential partners, particularly with dengue. Dengue, we will almost certainly out-license at some stage to a specialist pharma company. It's not beyond the pale that if we develop a Marburg and an Ebola program that goes through US FDA approval, and it's just a stockpile drug, that we could actually retain that drug and sell it, because you're not really having to assemble reps, you're not having to assemble medical affairs, you're not having to deal with the Sunshine Act, which for those who know the US is a pain in the ass. You're really just cranking the handle on a manufacturing run each time the drug expires. So I'm kind of in 2 minds as to what we do with that drug commercially. Given that we may be filing an NDA in 2026, we're going to have to start thinking about it sooner rather than later. And we'll be announcing how we're going to move forward. Well, fingers crossed that the due diligence piece reads out the way we want it to on Galidesivir. But assuming it does and we complete that acquisition, we'll be looking forward to discussions with the FDA around what they would like to see, whether it's going to be 1 more monkey study, 2 more monkey studies. There's a possibility we might have to do the monkey studies, get an approval, and also have a ongoing monitoring trial in the endemic countries in West Africa. We'll have to talk to FDA about that, but that probably won't slow down an approval.